In this last section of video, we are going to discuss the fate of ammonia. As we have discussed, after biological nitrogen fixation, or whether it's an industrial process, the nitrogen gas is converted into ammonia. And we've also seen that this ammonia is absorbed by the plant body to form into organic nitrogen. So we are going to understand how this process is happening. So at physiological pH, ammonia is protonated to form ammonium ion. Physiological pH means the pH that is present in the bodies of living organisms respectively, which is their cellular level of pH. Now, so this is what happens. This is your ammonia, and this gets protonated, means an additional proton is added and it has formed into the ammonium ion. Now this ammonium ion is not at all safe for the plant. In fact, it's very toxic and it should not accumulate in the plant body at all. It should be really removed out. So what happens is this ammonium ion, you have to either expel it or you have to use something else. You should use that ammonia to produce something else. What will happen in that something else? You're going to use that ammonia to produce amino acids. So how it is done, we are going to see. So there are two main ways for that. First one is reductive amination. In reductive amination, what happens? You have the ammonium ion which is formed, the protonated version. It reacts with an organic acid. In this case, it is alpha-ketoglutaric acid. So when they combine, what happens? There will be a reduction process, and who is getting reduced here? First of all, what happens is there will be dehydrogenation. The NADPH will lose the hydrogens. The ammonia will go to the alpha ketoglutaric acid and it will form glutamate, which is a amino acid. It will form glutamate, which is an amino acid. It will release hydrogen, water, and it will release NADP in the presence of the enzyme glutamate dehydrogenase. So here I have drawn the keto acid structure, because what happens is alpha ketoglutaric acid is a keto base. It's a ketone based organic acid, so we call it as a keto acid. Normally for reductive amination you need keto acids. So what happens here? First of all, in this NH4, the ammonia will come here. It will come to this position of the carbon. Okay, and there will be the COO minus should become into carboxylic acid. So you will get a structure ultimately like this: NH2 COOH and C double bond O. So this is my amino acid. So what will be left here? There will be a H2O that will be left, and because the H required for the formation of COH came from NADPH, it became into NADP. So this is what happens here in reductive amination, where a keto acid is reduced to form into the respective amino acid. Another keto acid is oxaloacetic acid. Oxaloacetic acid can also undergo reductive amination to form aspartate. Okay, so this is the first method. The second method is transamination. Sorry for that. In transamination, what happens? You already have an amino acid, you will have a keto acid. Now this amino acid acts as a donor. Donor for what? Donor for the amino group. And the keto acid, it acts as the acceptor. So what happens? The amino group from here, it will move all the way to here. Okay, so as a result, the donor amino acid becomes into a keto acid. This becomes into a keto acid, and the acceptor keto acid now became into a amino acid. So what is happening here? Basically, there is a transfer of the amino group from one amino acid to another amino acid. So we can put it in a very simpler way. Let us say this donor, my donor here is glutamate. Okay, and this keto acid is oxaloacetic acid. Okay, so what happens now? The amino group is getting transferred from glutamate to oxaloacetic acid. So what it will become? It will become, glutamate will become into the keto acid, which is alpha ketoglutaric acid, and the oxaloacetic acid becomes the amino acid, which is aspartate. So this is the second method, that is called as transamination.